Hello, today is April 23rd, 2013, and we are at the Boston Convention Center for Experimental Biology. My name is Jessica Bradley, and I'm a graduate student at LSU Health Science Center in New Orleans.
My name is Annie Whitaker, and I'm a postdoctoral fellow at LSU, and today we have the pleasure of interviewing Dr. Maurice Goodman for the American Physiological Society's Living History Project. Dr. Goodman is a longstanding measure of the APS and professor emeritus at the University of Massachusetts Medical School. His research interests include endocrine regulation of metabolism.
Dr. Goodman, welcome, and we thank you for coming to the Living History Project.
I grew up on Long Island in New York, first generation American. My father was an immigrant. I was the first one in the family to go to college, and so there was really no family background or guidance for anything. But as I was growing up, I was a very good student in school, and people just expected me to be doing something. And I wanted to do something academically, and there's no one thing that really came up that said you're going to be a scientist or you're going to study something in biology. But I had a growing interest in biology. I subscribed to Scientific American as a sixth grader and did all sorts of science projects at school. Things were quite different then, and there were not too many people that from our community even who went off to college. This was right after the Depression, really, and I grew up in the 30s and 40s, and right after the war was when things started to blossom somewhat. But I took part in the Westinghouse Science Talent Search way back in 1949, 50, somewhere around that, and just had a growing interest in and curiosity about things in general, and seemed to focus more and more on biology, although everybody told me you really ought to be doing engineering or physics because that's where the money's going to be. And who's interested in biology? What are you going to do with biology? But that's nevertheless what I did. And when it came time to go to college, things were very different then. We didn't take college tours with our parents, take a week off and go around the country and see what schools we had to go to. There was limited guidance, and I applied to all of the usual schools, and I got into all of them. The only school that I didn't bother to apply to was Harvard, really. But I was applied to Columbia and to Yale and to Cornell, and all of these sounded good, but they were big, and I really wasn't too eager to do that. But then a new school opened in Massachusetts. That was Brandeis. And it was not yet even accredited, but I didn't know what accreditation was about or why a school ought to be accredited or anything else. College was college. And it just seemed very exciting to come to something new and to do something new, and really had a wonderful time there with some wonderful professors who did help to steer me. This was the first real professional advice I'd ever been able to obtain, and one of my biology professors pushed me towards the Harvard School of Medical Sciences at the Harvard Medical School. That was after trying to push me into medicine. There was a great deal of pressure for me to go into medicine, and I really didn't want to practice medicine, and I thought I want to teach and I want to do science. And so that's how I ended up at the Division of Medical Sciences at Harvard Medical School, and that was the background. When I was at Brandeis, Brandeis was a small school and didn't have an awful lot of money, and so the science and the research that was there was really geared to inexpensive and small animals. So I did some research with Drosophila while I was there, and we had a wonderful microbiology department. Or not a department, but several professors in microbiology, and I was steered by them to, again, if I were going to go to the Division of Medical Sciences, I should go to the microbiology department, and I was accepted in microbiology as a result. But at the same time, we did have a physiology professor in a physiology course. Gjerding Olsen was his name. He was a wonderful friend and a wonderful, a wonderful friend and a mentor. And he was a student of Frederick Heisau's at Harvard. And that name should, I hope, mean something to you. His interest was in endocrinology, but it was also pretty broad in a number of other areas. And when it came time to graduate, Oli, as we called him, knew that Professor Heisau's lab had an opening over the summer if somebody wanted to go to work there, somebody being me. And he steered me to Professor Heisau, who was a giant, really, in his field. And I spent that summer working with rats, working with estrogens, and trying to understand how estrogens work. This was very primitive time. This was back in 1956, the summer of '56. We didn't know about receptors then. We didn't know about molecular biology. Remember, DNA was only worked out a few years earlier than that. So it was quite primitive. But I published my first paper as a result of that work that summer in Professor Heisau's lab. And from there, then school started in September, and I went on to the Division of Medical Sciences, which was a conglomerate kind of course. So we had there were 13 of us in that group at Harvard Medical School, and we had faculty made up from each of the basic science departments. And the representative for physiology was Ernst Nobel, who was a very charismatic and a wonderful, wonderful person that I will always be indebted to. And although I had come in really accepted by the microbiology department and was expected to work in microbiology, Ernie kept at me over and over again, and we became very close friends. And I would go out to dinner with him. He put the full court press on me. He had no graduate students prior to that. He was still an assistant professor, just really starting out. He had just published one of his major accomplishments, which was the species specificity of growth hormone. And I became interested in growth hormone, and little by little it became clear that I should work with Ernie. And I started working with him. And one of the other students in the medical sciences program was Julie Hotchkiss, who ended up being Mrs. Nobel a little bit later on. And the three of us really ended up doing things socially. It was somewhat improper, I guess, for a professor to be dating a student. And so Ernie would come to me on some occasions, say, why don't we go out for dinner? And by the way, will you ask Miss Hotchkiss to join us? So I was sort of a John Alden kind of character here. But that was a wonderful marriage, and they're both wonderful people who did lots and lots of good science. Anyway, I did my thesis work with Ernie, and the physiology department at Harvard was a very, very good place to be trained. It was very, very rigorous. Some really outstanding people there: John Pappenheimer, Cliff Barger, Gene Landis. You name them. They were shining stars. And as I finished my degree, the question came up, where was I going to go for a postdoc? Well, Ernie Nobel really was of the same lineage as Dr. Heisau. In fact, Heisau was sort of the father figure of a whole school of endocrinology. And Ernie had gotten his degree with Sam Leonard, who was one of Heisau's very early fellows. And Ernie had come to Harvard to work with Roy Griep, who was another one of HISAW's early fellows. And at the endocrine meetings, I got dragged along to dinner with these people. And one evening at dinner, Professor HISAW asked me, well, what am I going to do when I finish? And I wasn't sure at that point. I was thinking perhaps I would go to England and do some work in biochemistry there. And he said, well, why don't you get a kind of a fellowship that lets you do whatever you want? Pappy, as we called him, was then a senior fellow in a very elite, snotty group at Harvard, the Society of Fellows. And as a senior fellow, he had a great deal of say in who was going to be, which of the junior fellows were going to be selected. And thanks to my earlier association with him, I ended up being a junior fellow at Harvard, and which was a wonderful experience, mainly because I got to see him every week. We went there was a weekly dinner every Monday night. We had a weekly dinner, and with all sorts of exotic gourmet food. I mean, I ate bear steak, whale steak. You name it, we had it, and the wines were wonderful, and it was, and we often have guests, many of whom were Nobel laureates. It was just a very exciting thing, and the aim of the Society of Fellows was just do what you want to do. You presumably had some background and some qualifications, and now exploit that to the best extent that you can. Well, that went on for a couple of years, and while I was a junior fellow, I also worked as a postdoc in the laboratory of Ted HISAW. Ted Astwood, who was at the Tufts New England Medical Center, and Ted was a student of Fred HISAW's. I mean, this is very incestuous. And working with Ted, again, exposed me more and more to endocrinology and to the major figures in endocrinology of the time. And I continued to pursue the work in growth hormone, which was an outgrowth of what I had started in Ernie Nabil's lab. Well, my most favorite was going over the data. And we didn't have computers. We had an adding machine, a Monroe calculator, which I used to do statistics. And that was hours and hours. But still, it was exciting to know what was coming out of the lab. And I guess the least favorite thing I did then was to have to vaccinate the monkeys. Ernie's research was with rhesus monkeys. And it was often pretty messy. When we'd get a new shipment, this was before the ban on getting monkeys from India came along. This was just right after the monkeys had been used to make the Salk vaccine, and monkeys were readily available, and people were. But we used to get a shipment of perhaps 40 or 50 monkeys at a time, and they came in these huge boxes, about 6 in a box. They'd fly them over, and we had to catch them. They all had diarrhea and various other problems, and we would let them loose in this big cage and then have to come back, let them kind of settle down. Then we had to catch them again and inoculate them or vaccinate them or TB test them. And so we had to get in the cage with them, and it was, am I waving my hands too much? We'd get in the cage with them, and it was a bit messy. So that was the least favorite thing. But I can smile about it anyway. My first APS meeting, because I went to two meetings regularly. One was the Endocrine Society, and the other was APS. Endocrine Society was very small at the time. They did not have concurrent sessions. So everybody in the society went to every session. And the first meeting that I went to was I think it was in Boston in 1957 or 58. And then my first APS meeting was in Atlantic City. Federation met at Atlantic City every year in April. They met the week in which the 15th of April fell. And I gave my first paper there, and Jane Russell, who was an outstanding endocrinologist, first at Yale, and then she was down at Emory with her husband, Alfred Wilhelmi. She chaired the session, and she was tough. And Ernie scared the dickens out of me about all the questions she was going to ask me. Here I was a second-year graduate student. But it was very rewarding. Again, there were not all of these sessions. The meetings were much smaller, and you were asked to. Not everybody who submitted an abstract gave a paper. There was no such thing as poster sessions. You were invited to present a talk, a 10-minute talk, and there were perhaps 3 or 4 simultaneous sessions, but not in endocrinology. All the endocrinology was, every session was different. There were almost no symposia and no named lectures at that time. So it was quite, remember, 1957, which was the end of my first year of graduate school, is when Sputnik went up. And the powers that be in Washington got scared, and all of a sudden there was money for training graduate students. And there was an explosion, really, of scientific training and talent after that. I mean, I had an NIH fellowship that, I guess as an undergrad, as a pre-doctoral fellowship. But as far as numbers of graduate students, were very few. I think I was the 5th person to get a PhD in Gene Landis's department at Harvard. And again, there were 12 graduate students in the Division of Medical Sciences, and one who took courses with us but wasn't in the division. That's very different from what you see now. And there was no money in it for salaries. I think Ernie Nobile, as an assistant professor, was making $3,500 a year then, which went a lot further than it would now, but it nevertheless was not a princely wage by any means. But see, you did it because you loved it. And the whole notion of competition in science wasn't there. We were all kind of chasing the enemy, which was ignorance, and we were helping one another. It wasn't a matter, I'm going to get there first. The grants were starting to become abundant, and you didn't worry about the next grant nearly as much as you do now. Or at least I didn't. Maybe I was naive, and maybe I just didn't know better, but you didn't do something because you were going to get the next grant. APS has threaded in and out of my career throughout. Being elected to APS, which is what used to happen in those days, was an honor. You couldn't just, I don't believe there are any graduate students, so there was no student members at the time. You had to publish a couple of papers in the APS journal, and you had to be recommended by 2 senior scientists, and then you would be voted in. That's before science became big business. And you know, once the scientific societies started holding these enormous meetings and having huge administrative staffs and so forth, they needed the money, and so they opened it up, and more people were encouraged to join because they needed the membership money. But in those days, it was fairly exclusive. So it was an honor, first of all, and I think Gene Landis and Cliff Barger wrote the letters for me. As I had taken a teaching job at Harvard. When I finished my junior fellowship, I joined the faculty in physiology at Harvard. So that was the beginning of my association, and those early years I was asked to do to work on the editorial board of the American Journal of Physiology. And I guess I was so eager and gung-ho in those days. I used to write long reviews and spend hours and hours poring over everything. Don Fredrickson, who was then at the NIH, was the chairman of the publications committee. And as a 33-year-old, he invited me to become the editor of that journal, which I refused to do upon advice of counsel, my chairman and other professors at Harvard. They say, you're too young, you shouldn't be doing this, you got to concentrate on making your career going, you can't do all of these other things. And so that's what I did with the American Physiological Society in those days. And then it was an honor also to be named to a committee. And shortly thereafter, I was named to the program committee. And that was in early 1970s, I believe. I'd already moved to UMass, and I was chairman of the department, and there were all sorts of other things going on in my life. But I joined the program committee. And at that point, what the program committee did was to take all the abstracts from a given area, screen them, organize them into sessions. Again, there were very few poster sessions. Poster sessions came in just about then, or maybe a little bit later. And that's all the program committee did. But I'm a troublemaker. And there were a few lectures, and I wanted there to be more. I thought the general lectures were important, that kind of summarizing areas that were about to be published, or that sort of thing. And I became chairman of the program committee shortly thereafter, and really pushed the council, to come up with money to pay for speakers, which they wouldn't do, because if you were a member of the society, that was reward enough to come and give a lecture. But we wanted people who weren't necessarily that. And little by little, I think Fran Ganong was president at that time. And little by little, we pushed to expand that sort of thing, to get more and more symposia and more lectures. And at that time, noise was being made about the society was getting big, and maybe we needed to divide up into sections. And as program committee chair, I was invited to go to a meeting at Bar Harbor at Bodil Schmidt-Nielsen's place. Bodil became president that year. And Bodil and Gene Yates and Orr Reynolds, who was then the Secretary General, or whatever you call it, whatever Marty's job is, Orr's had then. And we met for a week then and came up with a plan to sectionalize the program. The rest of the society was gradually being made into sections. And so we organized the program efforts then as sort of an executive committee, program committee, and a general member. So we would have representation on all the different areas, say, and each section would have a representative. And that representative, in turn, would have his own committee for that area. And then we had a program executive committee. And that seemed to work very well. And I think it's expanded from that now. But so one of my major contributions to the APS was to chair the program committee for the next 3 years of that newly organized program committee, and to get more symposia, more general lectures, and that sort of thing. And meanwhile, money was coming in, and we started getting some endowed lectureships, and that all, each section ended up getting its own senior lecturer that they could invite in. I was very close to Cliff Barger, even after I left Harvard. And one of Cliff's passions was the Porter Development Committee. Now, Porter had been a professor at Harvard, and he really was the founder of the Harvard Apparatus Company. Among his other achievements, he was a tinkerer, and he developed all sorts of instrumentation, including chymographs, and that, I don't know if you know what a chymograph is. That's probably long gone before you were in science. But when Porter died, he left the Harvard Apparatus Company to the American Physiological Society. And in their wisdom, the Council of the American Physiological Society said, we're not going to run a company. What are you thinking of? Up until that time, the Porter money had been used to pay stipend for an elite Porter scholar. So when the society refused to accept the company, which I think they've regretted ever since, Cliff took it over and formed the Porter Development Committee. Cliff had a passion for trying to make the society and make science in general more accessible to disadvantaged groups. And from that, he first had the Porter Development Committee within the society, and its activities were funded by the Porter Foundation, which Cliff chaired. And then I came onto the Porter Foundation, well, I guess in the 80s, and served with Cliff on that, with a number of members of the former Porter Fellows even. And then when Cliff died, I took over as president of the foundation. And that, so I've had the ongoing interaction with Marty and the society. I was co-chairman of the society's Porter Development Committee, as well as chairman of the board of the foundation. And I got also involved in the society. I got involved early on when they started the teaching archive. I participated a little bit at that beginning, and it was just too much to work in all of these areas. But I continued to do editorial work for the society, and I ended up editing the endocrine section of the Handbook of Physiology. So I've had a lot of interaction with the society, eaten a lot of meals on the society's dime, but that's okay. I guess one of the most gratifying was we were able to work out what the molecular basis of species specificity was. And it turned out to be a mutation in one amino acid in the hormone and one amino acid in the receptor, which turned out that at the active site, it turned out these two amino acids repelled each other. They were both basic. And so growth hormone of non-primate species couldn't work in the primate. And it was particularly gratifying because I was able to present that for the first time at the retirement symposium that we had for Ernie Nobile, who had been my mentor and my dear friend. So that was a very gratifying bit of science. I think now you'd have to do things differently than we did then. Again, as I mentioned earlier, we didn't think about competition. We didn't think about where the next grant was going to come from. We didn't think about application. In fact, nobody patented anything in those days. And in fact, it was dirty almost to think that you were going to have commercial application to what you were doing. So it was a very, very different world. So I think if I were starting now, I would try to find an issue that was relevant to something that was societally important, rather than knowledge for its own sake, which was the Holy Grail in those days, I guess that's really what it would be, and I would focus on that. I would probably not have gotten involved in as many administrative tasks as I did. Taking over as chairman of a newly found physiology department and a new school, and that kind of blossomed into taking on all sorts of other jobs at that new school. I ended up being the dean and chancellor even for a year, so that I might have finessed all of that and just kept my nose to the grindstone, which is what Roy Griep had advised me to do many, many years earlier, and just keep your head down, do your best, and sooner or later somebody's going to notice. And I guess that's still good advice. My first graduate student was Fred Goldberg, who was a medical student, but became a medical student because of family pressure. Fred had been interested in physiology. He had been a Harvard undergraduate, been interested in physiology, and had even taken a year off and gone to Cambridge, England, to do a year of physiology there before he came back to medical school. And he got halfway through his first year in medical school and was very unhappy with medical school. And Cliff Barger, whose shoulder everybody cried on, advised Fred to come and see me. And I will be eternally grateful to Cliff for that, among the many, many other things that he did for me. But Fred came to work in my lab as extraordinarily bright and eager and very much self-starter. He pretty much knew what he wanted to do, and we did experiments together. And in fact, he published, I think, 5 or 6 papers from his thesis research, and my name was only on 1 or 2 of them. Well, you know, again, it didn't matter. It didn't matter nearly as much to have your name on. I mean, certainly he was first author. So he and Fred went on to become, well, I don't know if you know his work, but Fred discovered a proteasome. And I really, to this day, don't know why he wasn't included in the Nobel Prize that was awarded for the ubiquitin work. But he's still young enough, maybe it'll come. He's about 70 now. So that was my first graduate student. My last graduate student, let's see, who was the last? Probably Yael Schwartz was my last graduate student, and she joined my lab. She already had a child. She was a widow, had worked in science, very many aspects of science. She was probably in her 40s, and again, she knew where she was going. Was a hard worker. We had a lot of interesting discussions and locked horns on a number of issues, but we remain very close friends now. So it was a very different experience. She and my wife became very friendly, and she's now heading up a drug company. They're all different. That's the first thing I would say. And I would say, don't pit them one against another under any circumstances. What else would I say? I would, well, my own sense is that you lead by doing, not by sitting back. And the example you set and what they watch you do and see how you do things and how you think about problems, that's the most important thing. You left out teaching. You left out teaching, which has always been very gratifying, and teaching at different levels, but very gratifying. What has been my favorite part? I like them all, really. And I think they are all complementary. Even the administrative work you might do on societal committees. You meet people, you talk to people. You never know where the next idea is coming from. You never know what association is going to form when you meet somebody who may know somebody who may have done something. So that the more interaction you have with people, with other people in your discipline, or even outside of your discipline, the richer you become intellectually, and the more it supports what you do. So when you're teaching, you think about problems that you might not think about otherwise, and you always have to go back to square one. You have to start with the simplest and how would I present this? If I didn't know X, Y, and Z, what would I need to know in order to follow QRS or whatever? And you think about sort of basic aspects of things, and that enriches how you think about your research problems and how you think about lots of other things. So they're all complementary, they all fit together, and it'd be very hard to pull them apart. One thing I already said, keep your nose to the grindstone. Sooner or later somebody will notice, but young people today are too impatient for that. They're used to instant gratification, so I'm not sure how well that would work. But I would say also pick a topic that has societal benefit, medical application. That's where the continuing support is going to be. As far as the laboratory is concerned, I would say pick a laboratory where the guy in charge is in the laboratory and not behind a desk somewhere else. And no, I guess that's enough. I've enjoyed these meetings here at EB. That's, for both social and scientific reasons. I mean, after a while, you kind of develop a cadre of friends and in the business, and you look forward to seeing them. You may only see them once a year, but it's always nice to see them, and always. And this certainly has been a good meeting, intellectually expansive, in that you learn a lot, and you learn a lot about things that you might not think about or do otherwise. So although you work in endocrinology, you may go to a renal session, and all of a sudden, there are things in the renal session that, hey, they have some bearing on, or this guy is using this tool, and I could really use that tool. So this has been a very good meeting, I think, over all the years. You know, I didn't really think about that till after I retired. You know, you're so involved and so into it that or at least I didn't stop and think about that sort of thing. Or why am I doing what I'm doing? I am what I'm doing, really, and that's why retirement got to be very difficult. You know, when you identify with what you do, when you are what you do, when you don't do it anymore, what are you? So you go from a who's he to or from a who's who to a who's he, and that's kind of a downer. But after a while, you get used to it, and then it's okay. A lot, but I mean, I can't tell you what's happened in the last decade because I haven't been involved in grant review, but I was up until then. When I first joined the endocrine study section, I was the generalist in the endocrine study section. So we had a bunch of thyroid people and a bunch of gonadotropin people and a bunch of adrenal people, and I sort of was the generalist. And I got 30 or so grants to review for each session. And people, there were senior scientists on the study section at that point. They'd already knew who they were, and other people knew who they were. They weren't trying to impress anybody. They really wanted to do a job and help the grantee get funded or improve the science, so that maybe if they didn't make it this time on a rewrite, they would write a better grant. The last study sections that I worked on, other than I did an international postdoctoral study section also, that later than that. But the last time I was a guest on the endocrine study section, the attitude, first of all, there were young, very competitive people who seemed to be more interested in convincing their colleagues there that they were very smart and that the applicants were very dumb. So it was more about the reviewer than about the applicant, and I found that, well, there was a comment made. Well, he doesn't really know what this is, or he's saying this, but I know he doesn't know it because so and so told me this or that. So it wasn't a matter really of judging what was on the paper in front of them, but again, it was a matter of being more important in front of your colleagues, and I found that just really horrible. And that's when they were funding 20 or 30%.